Okay, good afternoon, everyone. Good afternoon. Thank you very much for being here for our Emerging Researcher Spotlight Series session number 2. My name is Annemieke Verenhorst, and I'm the Associate Vice President Research at the University of Manitoba. So I'm joining you here today from the Benedictine campus at the Apotex Center. It's a beautiful space, and it's home to the College of Pharmacy, among other units. And thank you for those that are joining us online. Please feel free to let us know where you're joining us from. We are joining you here from Territory 1. So let me begin by acknowledging that the University of Manitoba campuses are located on the original lands of the Anishinaabe, Cree, Ojibwe Cree, Dakota, and Dene peoples, and on the homeland of the Red River Métis Nation. As a member of the University of Manitoba community, I feel deeply honored to be part of research networks that are in partnership with Indigenous people in a spirit of reconciliation and collaboration. So the Emerging Researcher Spotlight series was created to shine a spotlight on researchers in their first decade of their career. And this series allows us to learn about the impactful research that is being conducted at the University of Manitoba. And we also hope that the Emerging Researcher Spotlight series will capture the interest of students to find their mentors and networks that is of interest to them. So today we will be hearing from 7 researchers, and they are on the screen here. They are from the Faculty of Science, the Faculty of Kinesiology and Recreation Management, and the Rady Faculty of Health Sciences. And when we talked about putting this series together and we invited researchers to speak, we asked them about keywords that describe their research. And for the 7 presenters here today, you can see the word cloud with the keywords. And so you get a glimpse of what we will be hearing about today. So in terms of the logistics, I will introduce each speaker, and then each speaker has 5 minutes to showcase their research. And then following the last presentation, we will open up the floor for questions and hopefully answers by the researchers. And so I would like to ask the researcher, once you present here, please go and sit and face the audience, because that would be for a better conversation. You may have noticed the refreshments at the entry there. Feel free to sneak out and get some more during the presentations. I hope you won't miss too much. But we also will have a little bit of a networking after the question and answer round, so you can stay around, have some more conversations with the researchers and other people in the room. And we hope that you have a delightful networking event. So with that, it's now my pleasure to introduce to you the first speaker. And the first speaker is Juliette Mammei. And she is a subatomic physicist in the Faculty of Science. And when we asked Juliette for some keywords, she mentioned that her research is about nuclear structure, fundamental particles, standard model, dark matter and energy, and electron scattering. So please welcome the first speaker, Juliette.
I'd like to thank the organizers for the opportunity to be here to tell you a little bit about my research. Hopefully you can hear me. Okay, so my research is quite fundamental in nature. I tell my students that physics is the study of the space and time in which we live. So that still gives me goosebumps. My motivating question has been, since I was a little girl, what are we made of? So I always knew I wanted to be a nuclear physicist in a certain sense. So if you see here, this is meant to represent a slice of American cheese. When I was a little girl, I used to fold the slice of cheese. How many people did that? Fold the cheese, you fold it again, and you get smaller and smaller pieces, right? So when I was a little girl, I wondered, what would happen if you kept folding the cheese? How small of a piece could you get? What would it be if you kept folding the cheese? Eventually, you'd get a molecule of the cheese. And then if you kept going further and further and breaking things up, you'd get individual atoms. Atom is a word that actually means indivisible, but it's a misnomer because we now know that atoms are made up of nuclei and electrons, and that the nuclei are actually made of protons and neutrons. Everybody probably has heard that. Have you heard that protons and neutrons are made of quarks? So it turns out that most of the matter, all of the matter that is around us is made up and down quarks and electrons. And currently we believe quarks are the smallest particles. The forces of these particles undergo are mediated by bosons and photons. And any of the physics that works here on Earth has to work out in the universe too. So that is a fundamental belief that we have in physics. So this so-called standard model of fundamental particles and interactions is described mathematically. Don't panic. This is the only math that I'm going to show for my presentation. And this is shown in brief. You need to understand a lot of math to understand what this means. But the standard model is incredibly successful at describing and predicting a whole range of phenomena. So it's very, very successful, except where it isn't. So when we look out at distant galaxies, we see a number of things that make us think that something is missing. In fact, we believe that only about 4% of the matter and energy in the universe is described by this standard model that I was describing on the previous slide. So one piece of evidence is the rotational rates of galaxies. When we look at the galaxies and we say, oh, we can see the light from the stars and we can tell how much mass there is, so the rotation rates of the galaxy should follow this curve here, the lower curve. But that's not the curve that it follows. It follows this upper curve here. So there's mass that is there that affects the rotation rates of these galaxies that we can't see. So we very creatively called it dark matter. Another piece of evidence is gravity can act like a lens. And so you see here objects repeated and reflected multiple times. They are doing that because there's some massive thing in between us and the object, but we can't see the thing that is causing the gravitational lensing. So that's another piece of evidence. So my research is an Earth-based experiment called the molar experiment, which will look at electrons scattering from electrons, and will measure quantities so precisely that if there's a deviation from the prediction, that tells us that there's a particle that we're detecting that we don't know about yet. So it's searching for physics beyond the standard model. Canada has a large part in this experiment, thanks to the researchers at U of M, myself and my colleagues. We're responsible for approximately half of the experiment, not all financially. It costs $40 million US, but we're responsible for the design and development of about half of the experiment. So to do the experiment, we need an accelerator that accelerates electrons close to the speed of light, so 0.99913 times the speed of light, so very, very fast. So we don't do that here. We do it in Newport News, Virginia at Thomas Jefferson National Accelerator Facility. You may have encountered an accelerator if you've ever had an X-ray. These are pictures. I have the sources here for all of the images, but this is a picture of Madame Curie, who actually took X-ray trucks out to the field during World War I to help doctors treat soldiers during World War I. The pictures on the bottom here are from a proton radiotherapy institute in Newport News, Virginia, that was started with the help of one of my colleagues, Thea Keppel. So not only can nuclear physics produce instruments that help us diagnose cancer, but it can help us produce instruments which help us treat it. And so because of nuclear physics knowledge, we know the distribution of protons in materials. Humans are mostly water, so the distribution of protons in water. And so we can target cancerous tumors while minimizing the effect to other surrounding tissues. So even though my research is highly fundamental in nature, the spin-offs and effects on society can be far-reaching. Thank you.
Thank you very much, Juliette. I will just walk here because otherwise nobody hears me. Thank you very much, Juliette. That was extremely interesting. So our next speaker is Yale Michaels, who is from the Max Rady College of Medicine. And Yale's goal is to develop better, safer, and more affordable cell therapies for cancer and immune disease. The following keywords give some perspectives of what Yale's research interest is all about: stem cells, synthetic biology, cancer, immunotherapy, and genomics. So please welcome Jill.
Great. Well, thank you so much for the invitation and for the opportunity to speak here. That was an incredible first presentation, and now for something completely different. So when we or our loved ones get sick, the medicines that we take generally fall into one of these two categories. So either small molecules in that cartoon on the top, or proteins, which we call biologics. And whether we take these medicines orally or through some type of injection, the goal is that these drugs will reach the affected tissues in our body and over time be able to correct that disease state. But the reality is that both small molecules and biologics are actually widely distributed throughout our body and tissue when we take these drugs, so not just going to the sites where they're needed. And this is a big reason why these medicines typically have side effects. Now, the other fact that we need to take into consideration is that as soon as we take these medicines, our bodies start breaking down and excreting these drugs. And so we need to repeatedly dose them over time in order for them to have their intended effect. And this can be a challenge both for cost, as well as compliance and pain when these medicines are taken via injection. And so in my research group, we're really interested in taking an alternative approach, which is to use human cells as living therapeutics. And the reason why we're so excited about this is because living cells actually have the ability to track to the specific sites of disease within the body, so avoiding those off-target effects associated with those conventional medicines, which can make them safer. And they're also able to persist in our bodies for years, or in some cases, even decades. And so the idea is for a chronic disease, we might only need a single dose of a cell therapy, which could last our entire lifetime. And so I want to start by giving you a real world example of what this actually looks like in practice. So I want to introduce you to a type of cell, which I think you'll hear about again when Deanna presents, called a T cell. So T cells are agents of our adaptive immune system, and their normal role is to patrol our body and look for virally infected cells, which they can actually potently kill. Now, one really interesting idea that researchers have had is to actually genetically engineer T cells to express these synthetic receptors, which we call chimeric antigen receptors, or CARs, so that instead of targeting these normal viral targets, that these T cells can actually seek out and destroy tumor cells. And so the way that this therapeutic works in practice is that a cancer patient comes in, they provide a blood sample, and then T cells are isolated back from that blood sample and genetically engineered, typically using some sort of virus, so they can express these synthetic chimeric antigen receptors, or CARs. And you can think of the CAR as like a lock and key mechanism that now allows these T cells to recognize, bind to, and kill tumor cells. So after these cells have been genetically engineered, they undergo some quality control testing, and then they can be infused back into the patient's body, where they can search out the sites of cancer and kill the tumors. And as it turns out, when these have been tested in the clinic, in particular for treating leukemias and lymphomas, these CAR T therapies have had really impressive results. So we're talking about now treating leukemias and lymphomas that have been refractory or resistant to frontline, second-line, third-line therapy. And in the case of these CAR T-cell treatments, over 2/3 of patients treated have a complete response. So this is really exciting in a space that's previously been quite difficult to target. And as a result of this really strong evidence in the clinic, there are now 6 CAR T-cell therapies which are approved for use in Canada and the US. So this is all wonderful news and really exciting stuff. But one thing that we need to think about is that these living therapies in general, and these CAR T-cell therapies in particular, are really expensive. And the reason for that is if we go back to those conventional medicines, these small molecules or these proteins that I talked about before, those are mass produced in large scale reactors. So we have economy of scale when we make those drugs. But when it comes to these CAR T-cell therapies, each time a cancer patient comes in, we're doing a one-off bespoke manufacturing process where every single time we're making a new medicine. And because of this, the manufacturer suggested retail price for these commercial CAR T-cell products is $500,000 for one dose. Now, our goal and our vision is to make these therapeutics accessible to anybody who needs them, not just people that are either independently wealthy or live in a wealthy country. So how can we do this? And rather than sourcing or getting these T cells from individual donors, one of the things that we're pursuing in my lab, and other labs around the world are working on this as well, is what if we can manufacture these CAR T cell products using a special type of cell called a pluripotent stem cell. So the reason we're so excited about pluripotent stem cells is you can take these cells out, they exist outside of a patient's body, and they're capable of growing, dividing, and expanding massively. And they can even be grown in large-scale bioreactors. So we think of this as being a renewable resource that can allow us to make CAR T-cell therapies and other types of living medicines. And so one of the challenges is to be able to take these pluripotent stem cells and actually get them to differentiate or develop into T cells outside of a patient's body. So this is one of the ongoing research areas in my lab, is to figure out conditions where we can get these stem cells to turn into T cells, which we can then use as CAR T-cell therapies. And our vision here is to be able to take these life-saving medicines and to drastically reduce the cost. So because the pluripotent stem cells are this renewable and self-growing resource, we anticipate that we can make these CAR T-cell products at about 10-fold less expensive than these commercial products are retailing for right now. And our hope is that in doing that, we can take these powerful living medicines and make them widely accessible. So I'll just end there and say thank you again very much for the opportunity to present here.
Oh, thank you again. Thanks, Yale. That was greatly interesting. So we are moving on. I think there's a little bit more similarity with the next speaker, at least between Yale and Ayesha. So welcome Ayesha Saleem from the Faculty of Kinesiology and Recreation Management. Ayesha is interested in cellular connections and studies how cellular connections change, for example, during exercise or due to the aging process or disease. And keywords that help describe the research program led by Ayesha are extracellular vesicles, pretty good, cancer, diabetes, asthma, and breast milk. So welcome, Ayesha.
Great. Thank you so much for the introduction and for inviting me to be a part of this excellent new series. And I really should have thought about putting all those keywords in, because then I had to show all of that data, but we're going to try and fit it into the 5 minutes that the other two speakers did a really good job of doing. So extracellular vesicles and mitochondria, the two loves of my life. As a graduate student with expertise in exercise physiology, muscle mitochondrial biology, I've always been fascinated by how exercise has these wide-ranging systemic adaptations. You get on a treadmill and you run, your muscles do the work, but your brain adapts, your kidney adapts, your mood improves, you have better sleep, you have improved fertility. Like, how is that happening? And that brings me to what I study in my lab, which I think are an excellent candidate to explain these systemic effects of exercise, known as EVs or extracellular vesicles. These are nanoparticles of information that are released from all living cells. So it doesn't matter if you're a potato or a human being, your cells produce these EVs. And the magic is that they can encapsulate biological cargo, so mRNA, protein, metabolites, DNA, minerals. And because they can do that, it makes them an excellent carrier to transmit information from one cell to another, from one organ system to another. And for me personally, makes them an excellent candidate to deliver skeletal muscle-derived adaptations that we see with exercise on a whole body level. So starting 5.5 years ago, the first thing we did in my lab was established an in vitro model of exercise. And I'm not sure if you can see it, but these are myotubes contracting in a culture plate. And this mimics the contraction that you see during exercise. And we can isolate EVs. And if we do this contraction for 4 days, we kind of get the adaptations in our cells that you would see in a human being in like 8 weeks. So you cut down that 8 weeks into 4 days, makes it a very effective model to study extracellular vesicles with muscle contraction. And of course, this work wouldn't be possible by all the collaborators and all the excellent students who've been working on these projects. This is what they look like. They're, like I said, nanoparticles, so smaller than the COVID virus that shut down the planet Earth a few years ago. And you can see our isolation system allows us to isolate and purify intact vesicles that we can tag and label and make sure that they're being delivered to the recipient cells when we're treating them. So what did we find? Something really, really cool. And I know I'm biased, but hear me out. We contracted skeletal muscle fibers, and we took these EVs, and we put them on lung cancer cells and muscle cells, and we saw totally different effects. In muscle cells, we see an increase in mitochondria, which is what we expect to see with exercise. So EVs might be transmitting these benefits of exercise to the muscle. But in cancer cells, absolutely no effect on mitochondria. But in fact, there was increased cell death, reduced growth, and all of the things you want to see happen to cancer cells. You don't want them to grow and get better. So this is really, really fascinating. We don't know how this happens. We don't know what is the cargo that is changing. Is it protein? Is it RNA? We don't know how things are getting inside. And we don't know if these EVs will have the similar effect if we put them on other cell types, if pancreatic cells or adipose tissues or cardiomyocytes. So lots of questions for the rest of my probably research career just from this slide, which is, what research is supposed to do. But speaking of mitochondria, that's something that really is a very important organelle. Like everything we do is because mitochondria exist in our body and they make energy. So if I'm standing here and contracting my finger, the energy to do that is coming from the mitochondria. If I didn't have mitochondria, it wouldn't work. So take a moment to say thank you, mitochondria. You do the work for us without even us asking you. Now, if you're unfortunate enough to be born with a mitochondrial disease, there is no cure. It can take up to 20 years for a diagnosis because it can affect a whole different set of organs depending on where the mutations are. So we're really interested in using EVs to use them as biomarkers of mitochondrial disease. In parallel, we're trying to establish the use of fetal EVs, so EVs coming from the fetus, in maternal circulation. And we're trying to use transgenic models to establish if this could work, because if so, we could potentially use fetal EVs to diagnose congenital disease like mitochondria while the mother is pregnant with the child. So work on this is ongoing. And speaking of child health, because I work at CRIM and because I'm fascinated by child health, we are also looking at EVs and how to promote optimal child development and growth, and collaborating with the Child Study, one of the largest population-based birth cohorts in Canada. We're looking at EVs in breast milk and using in vitro markers in vitro models where we are isolating EVs from breast milk from mothers with and without asthma and culturing them in human airway cells, or using cross-culture models of gastrointestinal barriers. We're trying to study how that affects child or recipient health cell function. And we're also collaborating with researchers in Texas to study airway contractility with these EVs from the breast milk from the mothers. So lots of research going on, basic, translational, clinical arms. I think I took it to heart when they said, as a PI, you can do anything you want to, and I want to do everything. So if there are graduate students in the crowd or postdoctoral fellows who are excited by this, first, seek help, because that's weird. But second, come talk to me, because we are looking for graduate students and postdoctoral fellows to join our lab. And speaking of the lab, I get to stand here and make it look all nice, but the work is done by the amazing students in my lab, 3 of them who are sitting here, an army of collaborators and mentors and funding agencies. So thank you so much for listening.
Thank you very much, Ayesha, and thanks to the students who will come here to support her. That was great. The next speaker is Sabine Kuss, who is a chemist in the Faculty of Science, and Sabine investigates molecule transport across cell membranes by electrochemistry. So we are continuing on the cell theme. And keywords to describe Sabine's research include bioelectrochemistry, biosensors, disease detection, drug resistance, cancer, and electroanalytical chemistry. So welcome, Sabine.
Thank you. Okay, hey, hello everyone. So I switched the microphone because I'm a mover. Yeah. Okay, so I'm Sabine Kuss, and my team is the Laboratory for Bioanalytics and Electrochemical Sensing. So what do? We transform electrochemical methods into viable, commercially available point-of-care devices that we hope that they can assist physicians to make better decisions in terms of diagnosis and treatment of diseases. Yeah, so how do we do that? We work in the field of bioelectrochemistry. That means we use electrochemical techniques and apply them to biological cells. So we study bacteria, we study tissue cells, we study cancer cells, and so on. So how does it work? We work with sensors that we hope or that give us information about biomarkers and indicators for such diseases. So what we do here is these sensors look like needles. Yeah, so one is shown there in the top middle image, and this is basically a metal wire sealed in a glass capillary. We can use these sensors and position them really closely to living biological cells. So you see here a cell membrane, and our sensor is right above it. What we measure is molecules that are being released from these cells and diffused towards our sensor where they are electrochemically converted. So we're doing oxidation and reduction chemistry. So these processes transfer electrons, and we are counting electrons and get information about the disease state, and any phenomena related to diseases such as drug resistance. The instrument that helps us to achieve that is called a scanning electrochemical microscope. And this instrument is unique worldwide as we have it in our laboratory. It is customized for the analysis of biological cells. So here on the left, you see this instrument as a scheme. On the right, you see it in our laboratory operated by 2 of our undergraduate students. So this sensor, this needle-like electrode, you see here on the left how it looks, and it is mounted onto a Z-axis positioner. This can approach a substrate or retract. And the sample sits underneath in a Petri dish that moves in X and Y. So we have all 3 dimensions to position this sensor in close proximity to these living cells. So what we are monitoring then is the flux of molecules from these cells. As an example, on the bottom right-hand side, you see an image of 2 cells located side by side. And the colors indicate the diffusion of glutathione, one of the main metabolites of a cell. Yeah, you can see how the glutathione is diffusing away from these cells. Yeah, and we collect this electrochemical image. So what can we do with it? We have shown in the past that we can monitor photosynthesis as it occurs. So we do this by incorporating an optical fiber inside our sensor. So inside our electrode is now an optical fiber that can irradiate cells at the same time as collecting electrochemical information. So we can irradiate algal cells and measure the production of oxygen. So we are monitoring photosynthesis as it occurs. We also put a lot of thought into it, how to make these measurements accurate, and we can now detect reactive oxygen species that play a huge role in the development and progression of cancer by electrochemistry. One of our most recent publications here on the right-hand side shows that we can measure a disease in newborn babies called cytochrome C oxidase deficiency. And this disease we can detect by electrochemistry. We can not just detect it, we can also quantify it. Yeah, so that doctors know what medication to apply so that these individuals can live a happy and fairly normal life. We can also now scale down this electrochemistry that we collect with our scanning electrochemical microscope to a smaller benchtop system. And we have shown that we can use electrochemistry to distinguish between drug-susceptible and drug-resistant cells. Yeah? And not just that, we can actually differentiate between different degrees of resistance. That has sparked interest by external investors, and we now incorporated a spin-out company of our group called Sensible Technologies, Inc. Yeah, so we hope to really translate these methodologies to the commercial market, to translate methods into technology. Yeah, so imagine you go to the doctor because you're suffering from an infection of some sort. And the doctor could tell you within minutes or even seconds which medication would work for you the best. This is what this box does. Yeah. Okay, so we are not just translating methods into technology in my group, we are also transforming young, talented undergrad, graduate students, postdoc into confident, award-winning researchers. Yeah? And I thank the funding agencies that carry our work and you for your attention.
Thank you very much, Sabine. And I think we all want the box that you showed on the previous slide that would come in very helpful. So the next speaker is Deanna Santer from the Max Rady College of Medicine. And Deanna is from the Department of Immunology, which is also housed in the Apotex Center, that the space we are in here now. Deanna's research looks at human immune cell responses against viruses and after vaccination. Research keywords to describe her work is interferons, immune regulation, inflammation, and antiviral. The floor is yours.
All right. This is working. Okay. Yes. Thank you very much. I'm honored to be part of this amazing group of scientists. Really nice to see such a wide range of exciting research ongoing here at the University of Manitoba. And so today I'm going to focus on my favorite immune proteins, which are called interferon lambdas, and tell you about how I think of them as a Swiss Army knife in our immune system toolbox. So why immunology? This, the Yale and I have one thing in common. We really love immunology. And this really started as an undergrad. We're just thinking about how does our immune system deal with so many different pathogens, so many different infections, but yet also can be detrimental, can cause autoimmunity, can cause asthma and allergic reactions. And so when I actually looked back from my career this far, you can kind of see my own word cloud from my papers that have been published. I've really focused on viruses. So how does our immune system fight viruses, in vaccines, also in autoimmunity, such as lupus, and then now we're also studying inflammatory bowel disease. And so the real goal is, if we understand how the immune system works, we can use that knowledge to then develop new treatments for patients. And so to really come back to viruses, which my first love is that, okay, you get infected with a virus, what happens? How do we fight those viruses? And so we talk about, first of all, our innate immune system. And so you can think about the very first thing we have is our physical barrier. So that could be our epithelial cell barrier, it can be a wall like our skin that's going to stop those viruses from getting in. Or you can think about it as a chessboard, right? So you're having all the different players that are coming together to protect the king. In that case, we have many different types of innate immune cells, such as dendritic cells or macrophages, that are coming together to fight to make sure that king is not destroyed. Or if you're a sports enthusiast like me and love football, you can also think about the opposing team being the virus that wants to score a touchdown. Do you need your first line of defense or that defensive line on plus all the other players working together to stop that virus getting in, that touchdown being you're getting infected with that virus. But then you also need to have adaptive immune cells. So those are the more specialized cells, like what Yale is studying with T cells. And that is basically, if you have that breach and barrier, what happens next? You need to have those specialized cells that can then fight off the virus. That could be from B cells making antibodies, could be regulatory T cells, could be these helper and killer T cells. And it's really all of it together that has to come together for the proper immune response. And actually seeing in the virus shown in red, so in the first hours are my favorite proteins, the interferons that are induced by recognizing that there's this foreign protein or foreign antigen from the virus. And then really it's them together with the adaptive immune system, as well as many other cell types. But you can see here that these immune responses take days, 7 to 10 days, like we talk about in the vaccine. But it's really this innate response in those first hours to days that we need to really fight off the virus. And so my lab is really focused on these proteins called interferons. And we can, not surprisingly, starting my lab in the pandemic, not surprisingly, we've worked on using these as treatments for COVID-19 in both phase 2 and phase 3 clinical trials. And nicely, we could show that the interferons could actually accelerate viral clearance against SARS-CoV-2 and acute COVID-19. But unfortunately, our immune cells can also contribute to and cause disease, especially chronic diseases. And so over the years, I've studied various different versions of this. But our lab, or my lab, is very interested in inflammatory bowel disease, or IBD, in both children and adults. And unfortunately, this is a chronic inflammatory disorder of the gut. There's no cure, and there's many symptoms that we're really just trying to figure out how can we use our immune system and target that to stop these symptoms. But unfortunately, we don't have a cure yet. But what we think is with these interferons, if we can learn, especially this one family, interferon lambdas, we think this could be a new therapeutic target, so we could actually help those patients suffering with inflammatory bowel disease and then use that as a new therapy that we can develop going forward. So coming back to the Swiss Army knife and these interferon lambdas, I kind of think of them as the underappreciated interferon family in the immune system because they're really only discovered in 2003. And so my lab is especially focused on the words in red. So we're very interested in how we can use these interferons as antiviral therapeutics. And yes, we have studied them in COVID-19 the past 3 years, but we're also knowing that we could use these at any place the virus infects a mucosal part of the body. So it could be lung, gut. There's also really cool work done by others in the placenta. And then also really how can we use these interferon lambdas to promote gut healing and anti-inflammatory responses. So really it is a Swiss Army knife because it's not inducing disease, but it's actually able to promote these healing properties as well as be antiviral. And that's really going to lead us to a lot of years of research to really uncover how these proteins work. And there's really too many people to thank. A few of my lab members are here as well, so thank you for coming. But amazing students and staff are what we call sister lab, is the Armstrong lab here. We really do a lot of projects together. Our amazing clinician collaborators, because all of this is human work, so human immunology, and many other collaborators, both here and elsewhere. And a special thanks to all the donors and patients and their families, because really you can't do any of this work without their donations. And then lastly, the funding from both here and elsewhere nationally, so we can do this work and really discover some new things in human immunology. So with that, yeah, let's take questions later.
Thank you so very much, Deanna. So next up is Athan Zovoilis, who is also from the Max Rady College of Medicine. Athan is in the Department of Biochemistry and Medical Genetics, and his research is best described by terms such as RNA, bioinformatics, genomics, and artificial intelligence. Please welcome Athan.
Thank you. Thank you very much also for the invitation. I'm honored and really feeling great being among people doing such great research. I'm going also to speak about the dark matter, like Juliette, but not the dark matter of our universe, but the dark matter of our genomes. My lab uses bioinformatics tools and develops bioinformatics pipelines for the understanding of what we call the dark matter of our genome, which is non-coding RNAs, and trying to associate them with aging-associated diseases. And to do that, we are applying many different approaches, but our major platform is next-generation sequencing, and we are trying to develop algorithms where we can use the signal produced by the sequencers in order to analyze and do high throughput analysis of biological data. And within this context, we are also supporting the bioinformatics core within the faculty and also in Cancer Care Manitoba for researchers that want to use our tools for their analysis. So what is actually bioinformatics? We can imagine our genetic information as letters literally written in a book. And just to give you an idea of the scale of information that we now have to handle, if one page, let's say a single gene, would be around 2,500 letters, we would need more than 1,200 books, 1,000 pages each, to include the information that we have just in one cell. And of course, the question is, how do we read all this information? So until around 20 years ago, we have very, very big sequencers. And these very, very big sequencers would actually need more than a day to sequence just one page. Now, we can actually do everything with a sequencer that fits here in my jacket. So this is a lot of information. And we cannot go ahead to analyze and understand this information without using computational tools. Of course, on the other hand, this develops new opportunities and could provide a better insight for a number of diseases. And my lab focuses on aging-associated diseases, and particularly on cancer and dementias, for which aging is a main predisposing factor. And particularly what we have been working on is not the around 2 to 3% of our genome that encodes for proteins, but the rest of this, the other 97 out of the 100 books, which until recently have been regarded as junk DNA, evolutionary remnants. But now we know that actually we do have a big amount of them transcribed into what we call RNAs. In a sense, this information is actually used. And it's no surprise that until now we regard this as junk because we couldn't read this information. But now that we can read this, and here is part of the Promethean sequencer we currently have here in Winnipeg, we can sequence this part of the genome, and instead of finding this as transcriptional noise, actually connect it with a number of diseases. And my lab particularly is interested on the part of our genome which is consisting of many repeats. And although it may sound interesting and actually unbelievable, more than 50% of our genome is actually repetitive. Multiple copies for which until now we thought that were just evolution remnants. And this has been a very long pathway, both as a researcher myself and in my lab. We are trying to understand how these repetitive RNAs, and particularly one class of them, is connected with aging-associated diseases. And what we actually found at the end of the first phase of this journey is that this is just the tip of the iceberg. Because until now, we have been speaking about letters and books, and everyone knows ACTG, but in fact, the alphabet is much more complex, both for DNA and for RNA. And those modifications that make actually a new part of the information and change the information are very difficult to read. And to achieve that, we have actually employed a sequencing platform, which is the big brother of that one here, and it's called the Promethean technology, where we can sequence RNA and identify this modification. And in collaboration with a number of groups here in Canada and abroad, we have formed a consortium that aims to understand how this modification, particularly non-coding RNAs, are connected with various diseases. And we are, as part of this consortium, developing these algorithms, and hopefully this will provide an additional piece in the puzzle of understanding this new type of information. That one, I want to again thank you for the invitation.
Thank you very much. Fascinating stuff. So our final presenter today is Brett Houston, who is the hematologist and clinical investigator at the University of Manitoba and Cancer Care Manitoba. Brett is in the Max Rady College of Medicine, and her research can be described by keywords such as blood conservation, supportive care strategies, acute leukemia, and meta-analysis. So please welcome the final speaker, Brett.
Can you hear me? Okay, this will be comparatively underwhelming. I've never heard of a quark, and I don't carry pocket sequencers, but you guys do, and you're very cool. So I am a hematologist, so I'm a blood doctor, and I treat people with blood cancers. And I'm going to talk a little bit about our research and how we hope to impact patients that we see in the clinic. So I thought I would start with an example patient. Mr. O is somebody that I would see regularly in clinic each week. He's 71. He's previously healthy. They're always recently retired. They've often recently come back from Arizona after a Manitoba winter. He went to his family doctor because he felt tired and he noticed some new bruising, expecting nothing. And his blood work demonstrates that his blood counts are low. And so he has the misfortune of coming to my clinic, and we diagnose him with acute leukemia. And so he goes from being a very healthy person who has many future plans to being a cancer patient with a very devastating diagnosis. And unfortunately, management for Mr. O is very time intensive. So he spends a lot of time at cancer care, receiving his injections. He's reliant on blood transfusions, whether that's red cells or platelets. He's always at risk for life-threatening complications that might pertain to bleeding or infection. And unfortunately, his survival is markedly shorter than what he was hoping for. So there's a lot of challenges with Mr. O's course, and I think the bottom line is that we need to do better. And research certainly provides an opportunity and a platform for us to learn from Mr. O and to improve the care for patients who are to come. Although, unfortunately, it's slower than we would like. And so our research program here at the University of Manitoba is looking at ways that we can try to integrate research with clinical care. So learn from patients like Mr. O in clinic so that we can benefit future patients, try and remove barriers that tend to separate research from clinical care, and develop a bit of a learning healthcare system. We're also hoping to move the needle forward by using innovative research methodologies, whether that might be patient engagement opportunities, novel methods of consent, or the use of platform trials. So where we're starting is the development of something called the MyeloCan Trial Network, or the MyeloCan Platform, which is a national platform trial led here out of the University of Manitoba, which aims to provide a perpetual trial or a trial infrastructure across Canada that we can then use to study and learn from patients like Mr. O who are suffering from different types of blood cancers, like acute leukemia. And so essentially what we're doing is this is underway, and we're hoping that if we create this platform infrastructure, then we can start interventions and we can add in subsequent trials along the way so that when patients come to clinic, they have the opportunity to participate in multiple different clinical studies or research projects and have the opportunity to seek new treatments. So for Mr. O in general, we have met with the patient engagement group, and we've established patient priorities for patients with different types of blood cancers. We have a clinical trial that we're starting looking at the use of tranexamic acid, which is a medicine to reduce bleeding in patients who have low platelet counts who are at risk for this. We also have other trials that we're planning to start looking at ways that we can improve infection and minimize hospitalization among patients who are receiving treatment for blood cancers. And so that might mean that in the near future, when Mr. O comes to clinic, he would be eligible for one or many of these interventions on the MyeloCan platform trial. So that might provide information about how we could optimize blood transfusion support for him, variations in terms of chemotherapy administration, as well as strategies to prevent bleeding and infection. And so that is all that I have today, and thanks again for the opportunity to present today.
Yes.
Yeah, so thank you very much. That was fascinating. Thank you all for your great presentations. So we're going to the question and answer round, and really anyone who has a question here in the room can raise their hands and I will walk to you. I like to have some exercise, so feel free to ask as many questions as you like. We also have someone to, someone over there, James. Yes, James. Thanks, James. James will be monitoring the YouTube comments, and so if you have a question online, please feel free to post your question, and then James will read it out loud. So with that, any questions? Panelists are also allowed to ask questions to each other. If you have a general question for all of the panelists, those are always fun, or if you have a very specific question for one panelist, that is fine too. Yes.
I'll start. I have a question for Yale. Your T cells, how do they find these tumors?
Yeah, so it's a great question. So the answer to that, the question is how do T cells find tumors? So the answer to that is evolution. So T cells have evolved to express these things called chemokine receptors that are basically ways of them understanding a GPS signal of where they are in relation to other tissues and processes that happen in the body. And so they're able to follow gradients of these chemokines that can be released from different tissue structures. So depends on the type of tumor, but their T cells are also really good at migrating very quickly. So they can cover and scan a very large area, which allows them to have contact with many potential candidate tumor cells. So they're basically walking around going, no, that's fine. No, that's fine. No, that's fine. Oh, wait, maybe a tumor cell. So it's a combination of their ability to sense these gradients and to move quite fast to surveil this area that I think gives them these properties. But it's nothing that we as researchers are doing to them. It's something that evolution has conferred these cells with.
Any other questions here in the room? Don't be shy. I can ask a question for Sabine to take revenge. So Sabine, I know you also use electrochemistry to look at small molecules in the environment. And what I also always wonder is about, there's so many different small molecules in the environment. And so how can you make the electrochemistry in a way that it's detecting that particular small molecule you're interested in?
Does that work? Yeah. Okay. Awesome. Yeah. Thank you, Annemieke. So yes, small molecules, that's something I did not speak about today, but we also do biosensor development for the agriculture industry, where we try to detect toxins, contaminants that are harmful for anyone who wants to consume grain that's produced in Canada, for example. So how do we make this specific, or at least selective? So some molecules that are electroactive have a fingerprint. So we can look at the electrochemical signature of these compounds, and which can be quite selective. So we can look at the signature and can say, okay, this compound has this type of functional group, for example. But that not always works, and not all molecules are electroactive. So we have means to modify our electrodes, the sensors that you saw, and by using nanomaterials, we can use aptamers, we can use antibodies, that capture the molecules of interest and then very locally converts them electrochemically. So that's one way to do it. So there's many strategies to functionalize these electrodes, to make them specific. We always try to keep things simple, but that's why today I only spoke about the unmodified electrodes, but that's certainly an option.
Yeah, thank you. Very interesting. I'm looking at the room again. I have a question there in the back.
I also have a question from Dr. Sabine. So I just wanted to ask regarding that I read a technique called as patch clamp technique, which was in electrophysiology. So it was like kind of similar. So like, what's the difference and how it's used in those things?
Yeah, thank you for that question. So patch clamp is a method that has been or was invented decades ago, and it allowed to measure molecules as they are being released from cells, for example, or to measure the potential differences of cell membranes. Yeah, the difference is quite simple. Patch clamp is in physical contact with these cells. Yeah, you basically clamp them or you suck them onto a capillary. Yeah, we are trying here not to be in contact with the cells. So we try to create a measurement environment, an experimental environment where the cell does not even know that we are looking at it. Yeah, because we believe that any mechanical influence can alter their response to stimuli. Yeah, for example, medication. We look a lot about, okay, we are treating the cells with the medication and we look at how does it respond. Yeah, and we want to not to be in physical contact, not to introduce artifacts, yeah, or alter their response.
Thank you. And one last question from Dr. Saleem now. So just wanted to know that I understand the concept of EVs in the isolated systems, like in the in vitro systems. But if we talk about the systemic systems, is it possible to identify the EVs coming from which specific organ? Like if we talk about the human body, like from the blood, can we find out this EV is being released from the skeletal muscle or let's say any other organ? How it is possible?
Yeah, that's a great question. In a human model, it's a little difficult. You can look at expression of specific proteins, but as long as your protein is not expressed in any other organ, then you can use that as a marker, and then you hope that it's packaged in your EVs. But not all EVs will have that, so there's that gray area. But with transgenic animals, that's very easily done. You can create reporter animals, or you can use the Crelox model that we're using to track where the EVs are coming from in our system. So yeah, but that is the challenge in a human model, to see where they're coming from. Because when you isolate them from plasma, they could be from any cell type, really.
Thank you so much.
Thank you very much for those interesting questions. I'm going to walk to the other side of the room. I don't know who was first. Someone is being honest, so.
Yeah, thank you. I have a question for Sabine as well. When you use those aptamers or antibodies, how sensitive can you measure those metabolites or whatever you're trying to track?
Yeah, another great question. So that depends a bit about, that depends on the matrix we're looking at. Yeah. So when we detect mycotoxins in grain, we need to consider the grain wash basically that we are creating. Yeah. And that determines our limit of detection or limit of quantification that we oftentimes achieve. Same applies to the medical sector. Yeah. When we look at, are we detecting in urine or are we detecting in blood? That all determines what the sensitivity will turn out to be.
Yeah, but can you make it then more sensitive than conventional measurements, like antibodies I can use in the ELISA or whatever? But I imagine your technology could be more sensitive, right?
Yes, because we are basically converting molecules locally. So we are trying to remove the background noise by making our sensors smaller and smaller, so that the background noise scales down as well, and then we can almost measure single molecules. So when we look at our SECM, the scanning electrochemical microscope, for example, we are counting electrons. We can basically measure how many molecules is excreted from a cell per second. Yeah, if it's a well-characterized metabolite that we are looking at. Yeah.
My question's for Dr. Houston. So I want to congratulate you, because I don't know that everyone understands the scale of trying to unify and have a living and learning clinical model. And I think we have similar interests in trying to develop this. I wondered if you could tell me if you're also measuring how the barriers are being reduced, and what's actually changing to streamline the processes, and not just the fact that a trial is available for every patient, but what red tape, so to speak, you've also eliminated in the process.
Thanks for asking a question I can answer. I have no idea what you guys are talking about. Let's be honest. I think that That's a really great question. I think that it's not like a static process, and I think that there's a lot of dynamic elements that we're using to try and increase research efficiency. I think part of the success is at an institutional level and trying to operationalize efficiency within clinic. I think part of it is at an ethics level where you look towards removing kind of artificial barriers between clinic and research. So that might mean integrating consent into the normal clinical process, or actually doing research at the clinical visit and not creating a separate research visit. Or it might mean using like routine collected data, like registry data or data that's available in the electronic record to capture patient events and outcomes, rather than creating a separate EDC or a data capture sheet just because it's called research. And so I think we would all agree that that's, inefficient and a bit silly, but that's kind of typically how research is unfortunately conducted in the clinical realm. So those would be kind of like some institutional barriers. We also are looking at creating a network across Canada so that there's already like 15 centers that treat patients with this specific type of blood cancer. So there's a critical mass of patients and investigators motivated to try and improve and incorporate research into clinics. And by creating a platform trial, it's a perpetual trial. So it's always there and it's always open, and contracts are already available. And so if someone were to come from Ottawa, for example, and they had an idea or a trial that they wanted to do in this patient population, it would just be a matter of adding it to what is already ongoing. So if you think of doing a trial as like building a house rather than having to build a new house every time, you could just renovate the room in the back. Is kind of what we're trying to do. So it's not perfect, and it is definitely a bit of a work in progress, but we're going to start here and work on it over time.
But it also allows the people that are sitting with you to potentially access those through correlative initiatives in a very streamlined manner as well. And so I think it actually ties in the entire faculty and the ability for multiple different groups to work with you and the resources you're creating. So it's a very big endeavor, and congratulations on that.
Thank you, Dr. Banerjee.
First come there, and then I'll come back. And were you first or were you first? Oh, there's another question. Too many questions now. We'll go here, then there, then there, and then there.
Thank you very much. My question is for Dr. Mammei. And this is in my field. I just get to read about it every once in a while. But I particularly like the example about folding a cheese in itself. And there's always that Reddit question that comes up every once in a while, that can you infinitely fold a cheese in itself? And when you try to do that, what happens? And the reason why I'm asking that question is how it relates to how you study subatomic particles. Because if you try to do that to study them, do you not also risk that implosion with subatomic particles?
Oh, not an implosion. But that's a very good question. So I was thinking about this when I was preparing this talk. That's an example I use a lot. But as you fold the cheese and you get smaller and smaller pieces, you need more and more energy to break it up. We can fold the cheese within reason with our hands, but then when we go to higher energies, it's electrochemical energies. And then to do nuclear physics, we need really, really high energy. So that's why we need the accelerator to be able to break up whatever it is that we're looking at into smaller and smaller pieces.
Okay, thanks. My second question is just out of curiosity. Do you think time travel is possible?
Time travel?
Yeah.
Personally, no.
Did you say no?
But I would be happy to be disproven.
Okay, thanks. Just curious.
Thank you for those questions. And I'm from the Netherlands, and whenever you start talking about cheese, I get really hungry. So.
Thank you. My question is for Dr. Michaels. For the CAR-T cell therapy, I have a couple of questions about that. So what is the success rate for that therapy? And at the same time, what happens when the therapy fails? Do they give another dose? And are there side effects? Do they need palliative care when they take that therapy as well?
Many great questions there, and I'll try to at least answer some of them. So I guess I could start with the success rate. So they've been these have been tested now for many different types of cancers, but most of the evidence in the clinic is for treating leukemias and lymphomas, and actually a particular subset of those which relate to cells from the B lineage, so B cell cancers. And to say something, the efficacy is different for each subset of disease and each type of CAR T product, but to say something sort of general about this, I would say for the 6 approved CAR T therapeutics, the success rate is somewhere between half and 2/3 of patients have a complete response. So that means that you can't detect any more leukemia or lymphoma at some point after they're treated. But for many of these patients, they do have a recurrence. So it's not, for everyone, it's not curative. For some people, it seems to be. So there's now 10-year follow-up on some of the first patients treated with CAR T that have no signs of disease a decade after they're treated, which is really exciting. But for the other subsets of CAR T, and I'm not a clinician, so certainly someone like Dr. Banerjee or Dr. Schweitzer or Dr. Houston could comment on this more, in more depth than me. But many of these patients progress to other therapies like a bone marrow transplant, for example. There are cases where people have been dosed multiple times with CAR T, and some people who didn't respond initially or have a short-term response may actually go on to respond to a second round of treatment. I don't want to go, I could talk about CAR T all day, all week, for months. I don't want to go too far into detail, but in terms of side effects, there certainly are some. The most common side effects are immune-related side effects caused by this massive onslaught of T-cell killing of tumors and producing cytokines, et cetera. So something called cytokine release syndrome is probably the most common serious adverse event associated with CAR T. And it seems like almost everyone who's treated with CAR T gets at least some degree of this cytokine release syndrome, which can be managed fairly well. Clinicians have figured, not me, but clinicians have figured out how to manage this fairly well for most patients, but it can require ICU stays, which is expensive and dangerous and unpleasant. So there's still a lot of room to improve the safety and efficacy of these treatments. It's kind of think of this is like the early days of the personal computer. Like we have this cool thing that seems to be really powerful, but there's tons of stuff to figure out of how we can use this more effectively. So exciting area to get into, I think.
Thank you very much. And I'm going to James. Oh, that's going to be a bit of climbing here, but maybe you can pass it to James. Yeah.
So I just have a question online, and this is in particular for Dr. Michaels. And this comes from a software engineer who's asking, what role does the private sector play in your research, and what opportunities exist for non-academics to help build tools to help advance said research?
So this question's from a software engineer? Awesome. Yeah, I mean, the private sector plays a huge role in these CAR T therapies, and I kind of envision this whole program as being a partnership, like many of these therapeutics, and I think you covered this nicely in your presentation as well. There's a partnership or a continuum where some of these early ideas are tested first in academic institutions, and then when it comes to scaling up and building out, that they can be transferred to commercial ventures. So there are a number of companies that offer CAR T now, but there are also, like, for example, here in Manitoba, we have a local academic program to be able to deliver CAR T to patients. And sometimes those programs have like an agility to be able to do things quickly and more cost effectively. So I think there's room for all entities in this continuum. And in terms of getting involved, certainly it's possible to get involved in CAR T in the private sector. And actually software engineers are in high demand in this space because we're starting to think about, I say we, I mean the field is starting to think about how do we predict who's going to respond to CAR T and what the side effects might be. And computational tools are extremely important for that. So I think there are tons of opportunities, and whoever the person is that asked the question is certainly welcome to connect with me via email to discuss this more.
Thank you very much. Question there, and then I'll come to you.
Yeah, I have a question for Juliette. So what would you speculate is the dark reason for it?
Oh, that's a very good question. So by nature of doing the experiment that I'm doing, I think dark matter is something that will interact via the weak force. And that is what we see is electromagnetics. So the light from the lights is electromagnetic radiation. When we look out in space, we're radio telescopes, and that it's all electromagnetic radiation. But the weak force was discovered relatively recently. And a lot of my research involves using the weak force to study subatomic nuclear properties. So I can't say more than that right now because we really don't know what it is. There's lots of searches for dark matter particles. There's people studying in Antarctica. There's sensors that are being sent down into the ice that are looking for to intersect potential dark matter particles. If there is dark matter, if there is really a particle, it's here too. We just don't interact with it very much. So I saw someone speculate that there could be dark matter entities that are, cohabitating space, but we just don't interact with them because we don't interact using the same forces. So it was kind of interesting along the lines of the time travel question.
So my takeaway from that is time travel might not be possible, but Jedi might be real. My question is for Athan. So for someone who works kind of in the 3% of the known genome, what really made that possible was not only the technology to get the letter code, but also the reference against which you can compare the new sequence to understand differences and then ultimately make an inference on that. Given that for sort of the 97% of the genome, we have very limited reference material, particularly when we're looking at very rare variants and methylation patterns, etc. What is being done on that front in terms of overcoming the baseline reference knowledge, or how do you standardize the creation of new knowledge in that space?
That's a great question, and the truth is that the reason that until now we have not been able to study is because it's such a big amount of information that we couldn't access. And that's also the reason why until now there has been a lot of debate whether, for example, the RNAs that are produced there are still a transcriptional noise. So I would say now that we are at the last stage of this debate. So initially people thought that they have no function. Now, because there has been an increasing number of RNAs that have been shown to produce very strong phenotypes and be associated with certain diseases, now we are start building a database of RNAs, how this would look like if they are to be associated, for example, with a disease. But we still have a lot of work ahead. I mean, even from that non-coding RNAs that we have tested, many of them are kind of canonical non-coding RNAs, so they usually have a poly-A, they're spliced. So we haven't really started looking for those non-coding RNAs, for example, like chimeric RNAs. So there are RNAs that are produced by two genes, which are different chromosomes, and no one knows how those are made. And those chimeric transcripts are already known since 2005 and through the ENCODE project, but still no one has started working on them. So we literally know nothing, or for circular RNAs. So it's more that we haven't yet had time to dive into this kind of research, rather than, and of course we didn't have the tools to dive and study them.
Yeah. Any other questions from the audience or online?
I have a question.
Oh,
sorry. I can just. What would happen if you delete all of that non-coding DNA? Like, does the cell die? Like, the mitochondrial genome doesn't have any introns or junk DNA. It's like efficient, and I completely believe the junk DNA that we call it is doing something. We just don't know what it is. But has anyone tried that? Just deleting all of that?
As far as I know, they haven't been able to do for the big amount, but they have, for those knockouts that were made for a number of non-coding RNAs, they have actually had 3 cases. We have classified them into 3 cases. One is that they did have an important phenotype when you delete them. I mean, could be
lethal already from embryonic states or shortly after that. Then they had cases where they didn't find anything, and they have assumed that maybe there are compensatory mechanisms, and those RNAs, because they're not highly conserved, may serve a function, but there may be something that makes up for their difference. And there has been an increasing number of cases where initially they didn't find anything, but then when they tested in another context, for example, during response to cellular stress, during anoxia, they could see a very strong phenotype. So what seems to be the case, at least for encoding RNAs, is that because they're not very highly conserved, I mean, some of them you find them only, for example, in human or in mouse. They have been highly specialized for specific functions. So you have to nail down to find which exactly that function is.
So does that mean you're going to write a research grant together? And yeah. Any other questions for the room? Yes, I see. Oh, I see. We'll first go there to the audience. You can talk to each other while doing that.
Thank you. I want to take all speakers firstly. So my question for Juliette, and you mentioned about the subatomic particles, and as far as I know, if you want to understand subatomic particles, we need to understand first quantum mechanics. So there was a discussion in 1940s about quantum mechanics, and as far as I know, Albert Einstein denied quantum mechanics. So everyone accept that Albert Einstein has a huge brain and a huge impact on the physics. So, and also he said God doesn't play dice with university. But last year, Nobel Prize received quantum mechanics, and Albert Einstein failed on his opinions. So I just wondering that what is your personal opinion about it? And my second question is, what should we learn with this discussion?
So my personal opinion of Albert Einstein?
I know, I just
or
Albert Einstein failure.
So
actually,
yeah,
about
so that is one of the things that I think he was not right about. I forget who said it, but the quote is, "If I have seen far, it's because I've stood on the shoulders of giants." So quantum mechanics wasn't as much of a leap. For those of you that have seen Quantum Leap, it wasn't as much of a leap as maybe we're brought up to believe. Classical mechanics was developed very far, and then quantum mechanics is just a relatively small step, which is only that there are quanta, that there are photons, that energies can only take certain values in atoms. So yeah, poor Albert Einstein was wrong about that one. And you're right, it was proven recently with Bell's theorem, and it did win a Nobel Prize. So what was the other question? I forgot. I think what I tell I'm teaching a Quantum 2 class right now. And so what I told my students is you have to learn what we think we know so that you can tell if there's something else, if we're missing or if we're right, or we could be wrong, right? So you're learning the dialogue as it stands now as a student, but I don't know, biology when I was in high school was nothing like what biology is now. That would be my advice to you, is that learn what we think we know now, knowing that that might not be how it really is.
That's great. Thank you very much. So even Albert can be wrong. Would you still like to ask your question?
Yeah, a question for Ayesha. So it has been shown that exercise can help also delay significantly neurodegenerative diseases. So my question is whether the extracellular vesicles could pass the blood-brain barrier and implicate any role for them in their pathogenesis.
Yeah, great question. I mean, they can. That's the short answer. There are people who are trying to study that communication because the beauty about extracellular vesicles is that they're inherently produced. And in your house, you can go to the attic, you can go to the basements, no one's stopping you. So if something is inherent to us, they can pass through those barriers. And that means that we can leverage them to deliver drug or therapies in areas that are probably have been traditionally harder to access, or using any kind of AAV mediated vectors could have caused side effects. So we are not looking at blood-brain barrier in my lab, but we're trying to see if the fetal EVs are crossing the placental barrier. It's been quite challenging, but very, very cool and very interesting. It will make sense if it is. That is one of the central mechanisms of communication across evolutionary biology. So yeah, very cool. Thank you.
See question there.
I have a question for Deanna. So in oncology, over the last few decades, we've periodically come back to the immune system. Just when we think we have taken a step, we move away because we understand it less. Right now in cancer therapeutics, immunotherapies as a broader class, with cell therapies being a subset of them, are again in vogue as they've made huge strides in terms of improving patient outcomes. But we still have fairly crude approaches, and part of the reason we have fairly crude approaches is that we really only evaluate their biology in a static manner, often kind of at presentation, but nothing sort of as a real-time assessment of how the immune system is evolving or adapting. So a general question is, given the complexity of the immune system, where do we stand in terms of the usability of tools to understand fluctuations of interferons or other even immune cell compartments on a real-time basis, that we could understand the trajectory and then respond to those trajectories and make informed decisions based on those trajectories?
Yeah, okay, that's a very complicated question, but yes, so I mean, it's kind of like the same thing with all the things we've been talking about here, like exercise. There's so many different things that change how our immune system is, even daily, the microbiome, things like that. So really, unless you're doing a more longitudinal like assessment, so like we always do a snapshot, right? So we'll get blood from a person on that day, but we may not get exactly the same response even a month later. So we're even learning even males versus females, for example, right? So maybe we should be asking, what part of the cycle are they in? Like we're finding more and more that it's much more complicated than we originally thought. And you can't really get at that using mouse models. So that's why my lab is really focused on using human systems, even though it's more challenging. We get, biopsies and blood and tissues, but really it's still going to be a snapshot, and that is always going to be the limitation. But, we can't, probe people too many times. So the more patients we can study, that's really the only way we can get away with, not being able to get those longitudinal samples. But at least seeing before and after a drug treatment, like one of the collaborations with Versha here, we can actually see how that immune system, at least part of the immune system, changes with and without those treatments. But I agree, it's very challenging.
Maybe we need some electrochemistry in the body, so get it real time. So any other questions? No, I think that's a good time because it's 3:30. So I would like to thank everyone, particularly the panelists. This was wonderful, and I'm so amazed by the large teams that you're working in. So thank you for all the hard work and everything you do, and it's cutting edge research for sure. So thank you very much. So this concludes our second Emerging Researcher Spotlight series. Please stay around and have some more nibbles and drinks that are there. So I hope to see you again. So our next event is scheduled for March 28th at 9 in the morning. So it's for the early risers. It's in the Marshall McLuhan Hall at the University Center, Fort Garry campus. If you don't want to wait that long, you can also go a little bit earlier to see another event. And the good news is that that's tomorrow. And tomorrow we have the Knowledge Exchange. That's at the Degrees Diner at the University of Center as well, at Fort Garry campus. The Degrees Diner will be open during the event, which is from 7 till 9 in the evening. And we are going to talk about heart health and you. So we have Dr. Laurie Kirschenbaum, who is the moderator, and we will have a number of panelists there as well. So if you're interested, please come out. And thank you so very much for your time, for being here in person. Thank you to those that joining us online. Very much appreciated. And I will hope to see you in the near future. Thank you very much.